look at biotechnology and human intervention, so that's why we should watch that CRISPR video to start with. Um, and so this is just, that's canola, okay? That was actually genetically engineered right here at here in Saskatchewan at the U of S. They were able to take a mustard seed and yeah, yeah, at the U of S there. Invented it. Um, um, what's that? No, no. They wanted something to make, like they wanted a seed that was high in oil content, so they could crush it and make oil with and everything. So they were kind of playing around looking for like a cold weather resistant mustard seed, and they just kind of came across this and went from there. Yeah, now it's a multi-million-dollar crop for the Saskatchewan, right? So this other one's just a cartoon of, or just a internet photo of a, you slice an orange in half and there's kiwi inside. Okay. So one of the first forms of bio. Okay. So yeah, absolutely. Race horses. Yeah, it's like a dog, if a not a dog, if a horse wins like the Kentucky Derby or something, millions. Yeah, yeah, just a stud afterwards. Okay. So yeah, they because they have these desired traits. They're a fast horse. All right. So things like juicier berries, rust-resistant wheat, um, all sorts of things. Corn with more kernels, etc. Like corn came from grass pretty much, and we were able to genetically engineer it by breeding it for desired traits. Yeah, it did, but we still got there. They didn't have like labs and microscopes. So breeders want each plant or animal to consistently have the desired characteristics. This might take many generations of breeding to get this right and can be done through artificial insemination. I think that's what most cattle breeders even do now, is they inseminate like in a lab and then breed the cattle. At least that's what I remember from the George boys telling me. Um, Now, another form of, so with selective breeding, you can get inbreeding as well. Sometimes it's known as line breeding. So this is just like with inbreeding, it's a controlled form of breeding, which crosses closely related individuals over a number of generations. And the reason they're doing this is because they know this line of people or animals or whatever it was has the genetic traits that we want. Oh yeah. Well, not mutations, but what? Yeah, like what ends up happening is, so the intent is to keep like your parent lines or the bloodlines pure. So what happens is it has some recessive genes that could be harmful to the organism that were masked by dominant genes in the past, could show up in greater extent during inbreeding. Oh yeah. Oh no way. Okay. Yeah. So like you're basically playing with a shallow gene pool, right? So there's more opportunities for those undesirable traits to come out. Where you want all this genetic variation and diversity, suddenly you're cutting that down quite a bit. Oh, that's kind of like royal families for generations. They said have like kind of kept things within the family to keep the bloodlines pure, but I don't think that's happened for quite a while now, right? So, and every once in a while you hear about like I remember in the news a couple of years ago, there was like this really isolated village in China, and they were encouraging like young people to come move there and like marry people in the village because they were all so far isolated, they all were kind of related. If you can catch my drift. They were worried about inbreeding going on in this village, so the Chinese government was offering money for people to travel out there. Not quite like that, but also like things get a little crookeder, and IQ's a little lower, and, you know, stuff like that. So now another form, instead of inbreeding, we crossbreed animals too, or sometimes. Kind of, but we get a little more um, varied in what we're doing. Okay, so these are individuals of different breeds, species, or genre are crossed. Okay, so examples would be like ligers, a jag lion. Yeah. Now, and they're sterile. Okay, they can't reproduce, just like a mule. When you cross a donkey and a horse, you get a mule. They're sterile. All right. Yeah. Yeah. What's mules can? No, they can't reproduce. No. Yeah. No. It'd be nothing. Yeah. No. Because they're two different species, so they don't have the proper chromosomes to match up. I think a donkey has more chromosomes than a horse, or the other way around. So their genetic offspring, because of that, are rendered sterile. No. There is one exception. It's called the American red wolf. That was the one that I could find. This is a cross between the gray wolf and a coyote, actually. Yeah. There's an exception to this rule, and is now a hybrid species. Okay. This one can. Most often than not, they can't, though. Okay. Yeah. Well, yeah, they're both like canines, but yeah. So the offspring of plants or animals in crossbreeding are often called hybrids. Most individuals resulting from crosses will be sterile, but some will show characteristics called hybrid vigor, in which the offspring tend to be stronger or more vigorous than either of their parents. All right, namely plants. Okay. You would absolutely. I was just going to say that. Yeah, they're yeah, like they were bred like I don't know why I know this, but when like settlers were coming west through the states, they all took mules because they were strong and were like could go forever. They were like yeah, mules are like what brought settlers. You know, I don't know why I know that. Yeah, and they could just endure forever. So okay, okay, biotech in action. So so polyploid. These are cells or organisms that contain more than one pair, more than two paired sets of chromosomes. So homologous pairs. So most eukaryotes are diploid. They come from haploids. Remember, you get half and half. Most prokaryotes, so like viruses and bacteria, are haploid. Polyploid types are labeled according to the number of chromosomes. So you can have things like triploid, so they have 3 sets of homologous chromosomes. Tetraploid, 4 sets. Or hexaploid, 6 sets. And this is where you're going to get different things like, so with three like triploid organisms, an example of that would be seedless watermelon. What's that? Oh, a slice of watermelon. Fair enough. Without seeds. Yeah. Tetraploid, salmon and trout. Okay. A type of fish. And then wheat and kiwi would be your hexaploid. So you see here would be a diploid organism. And then so like they have 3 sets of chromosomes, so they have 6 chromosomes. Here would be a tetraploid. There's actually 4 sets of chromosomes there. So there's 12. Okay. So polyploid plants are usually larger, hardier, faster growing, and generally more desirable than their normally diploid parents. This is also a common technique for overcoming sterility in hybrid crops. Tricattail is a hybrid of wheat and rye. Hybrids are sterile. Did I get that right? Triticale? Farmers out there, you guys heard of that one? Triticale. Guess not. Okay. So it's a hybrid of wheat and rye, and its hybrids are sterile. After polyploidization, the hybrid becomes fertile. All right. This is induced by treating seeds with a chemical which inhibits chromosome segregation during meiosis. So it's almost like they spray them with a plant hormone to promote fertilization like this. So sometimes polyploid crops are sterile, and this is preferred. Seedless varieties of fruit, like seedless watermelon, they're sterile, which is great because you don't want to spit out seeds all the time. New plants are created by using asexual techniques as cuttings. So they'll just use cuttings. Sometimes what they'll do is they'll even use more hormones to promote fertilization with flowers. So here's some example of polyploid organisms. So this is an octoploid strawberry versus a diploid strawberry. See how much bigger it is? Okay. Yeah, that's what they do. That's how they get them to become so big. Yeah, there's nothing to them. Yeah. Yeah. Then this is just an example of diploid daylilies versus tetraploid. You can see how much more offshoots they have. And then bread wheat, it actually has 6 sets of chromosomes in it. It makes it bigger, more desirable for grounding up into flour. Okay, now the next type of biotechnology that we're going to look at is genetic testing and screening. Okay, so we talked about karyotyping the other day, and we talked about chromosome deficiencies. You can actually test a fetus for genetic disorders if parents know they are carriers to determine if the child has the disorder. Or they do genetic testing, I think, all the time, if you want, for free, for certain things like Down syndrome and stuff like that, the big ones. And it's just with a simple blood test. If you're using things like in vitro fertilization or IVF, an embryo can even be tested before it's implanted. Yeah, go ahead. You can also be tested as an adult to determine if you have a genetic disorder. You can actually now get all the different tests, and they can run and check you for a lot of different things now that we've mapped out the human genome so much. There's still not lots that we know, but like you can be tested for different markers and stuff like that. Maybe not to indicate that you would totally have a disease, but you would have like the genetic disposition to potentially get that disease. Like different celebrities come out with it all the time. Like, what's his name? Thor. There, Chris Hemsworth, Dunedin's hero. You got a little upset with me when I didn't know his name. He's been what? It's been shown that he's at a genetic disposition. He carries some genes that could lead to Alzheimer's. So they're doing a bunch of stuff on him to help lower those possibilities. Who else? Yeah, I I can't remember. Yeah, she's got something too now. She's got some kind of disease. Um, there's an actress. Yeah, like Angelina Jolie. She's has a 90% chance of breast cancer or something like that. So like those are the big ones that they can really test for right now. It's generally the bad ones that we try to find right away. So and then what they can do is a bunch of different things. So steps following a DNA sample. So what they do is they'll remove amniotic fluid, blood, or saliva from somebody. For most embryonic, if you're testing a baby for genetic disorders or anything like that, genetic testing, they don't even need amniotic fluid anymore. They can just do a blood test from the mother. Yeah. So it used to be they'd have to like, yeah, yeah, like get some of that amniotic fluid. Yeah, it wasn't pretty. But now that's all changed. So what happens is they use restriction enzymes, kind of like the CRISPR ones, are produced by certain bacterial cells that bind to specific DNA sequences, and they'll actually cut the DNA at that sequence. So they chop it up. They use these to isolate specific genes or regions of the genome. Isolation of fragments through gel electrophoresis, which we talked about the other day. Remember, you put the DNA into the wells, use the electricity to pull them through. Compare the DNA with a disorder. So what they'll do is like they'll take the known DNA of someone with that disorder and they'll put that through the gel electrophoresis first. And okay, if it looks like this, then we know that they have, I don't know, ALS or whatever. And then they do that with your DNA and then they compare them. And if they're the same, you got problems. You're going to have issues. Okay? So portions of the gel containing DNA can be removed for further study. Cloning of DNA through a PCR reaction, okay? Placing DNA fragments, DNA polymerase, at 4 different bases and 2 short primers into a test tube to make millions of copies of a region of the DNA fragment. Okay, so what they'll do is with they'll actually put a bunch of your DNA into a test tube, and with DNA polymerase, remember that's the enzyme used to copy DNA. And what they'll do is they'll actually make copies of your DNA. And then they'll run that through the gel electrophoresis, okay? And they'll kind of compare where your base pairs line up as well. Okay, now this one's kind of cool. This was kind of the bee's knees before CRISPR came along. Recombinant DNA. This is a type of genetic engineering. It involves cutting a DNA sequence, either one gene or several genes, from one organism into small sections and inserting or recombining the sections in a host organism of the same species or different species, okay? So you can actually take DNA from one organism and splice it into another, okay? This is what gene therapy can be used for as well. These transferred genes would then carry out the normal effects in the new cell. So, for example, insulin-producing genes have been inserted into E. coli to make synthetic insulin for diabetics. So they used to like get insulin. They had to like harvest it out of organisms and stuff like that. Now what they can do is they actually put that DNA to produce the insulin into E. coli. And then the E. coli, because the E. coli is a virus, right? and it just goes and it goes and it goes. It produces all of the insulin we need. That's synthetic insulin now. Okay. Yeah. Yeah. They get this bacteria to do it for free, and then they charge you out the wazoots. Yeah. And make. Well, yeah. That's what pharmaceuticals are. Oh yeah. Yeah. Mhm. So don't get diabetes. Uh oh. Uh oh. Good luck to you. Um. Get a good. Yeah. You live here. Yeah. Get a job with insurance. Get coverage. So that's one example. Goats have been engineered to produce a protein that prevents clotting in blood, and they actually use that in surgeries because you don't want blood clots while you're operating on somebody. You don't want that. Other examples, they actually have taken like jellyfish DNA, which glows in the dark, and they can put it in things. We did that for. I used to do that as a lab. We would take E. coli, and we would add the jellyfish DNA into it, and then we would grow the E. coli. What's that? We don't have time because we're going to do extra dissections this year. But I can show you photos of it. And like, yeah, you can actually, like, we'd smear the E. coli on the agar just like we did in health science. And then you'd let it grow for a little bit, and then you take a black light, and the E. coli would glow in the dark. So it works pretty well. So they've been doing that with other animals. Like they'll put jellyfish DNA in like plants and stuff like that, and plants will glow in the dark. All these different things. Okay. But E. coli tend to be the ones that we use because if we want something produced, E. coli reproduce quickly, and they have a high metabolism, so they just keep pumping things out all the time. They actually get E. coli now to make vegan protein for people too, because most protein comes from animal sources, so they can actually get E. coli now to produce animal-based proteins for people. For vegans, really. It's kind of wild. I was reading about that the other day. Huh, indeed, isn't it? All right, now cloning. So a clone is derived from some part of the body of another individual created from differentiated cells. So basically what I'm saying here is genetically, clones are the same in chromosome and gene composition as their parents. So it'd be like me taking skin cells from Janelle and then using them over here, away from Janelle, to produce a second Janelle. You were just there. Oh yeah, like yeah, Dolly the sheep back in like the late 90s. Yeah, and people have cloned, like scientists have cloned monkeys. There was an Italian scientist who claimed to clone a human, but that was completely wrong. um, with the like, it doesn't work out well every time, even with the animals. Like we'll get into that here. Yeah, like they have short lifespans and a lot of health issues. I think I have that on a slide. No, Dolly didn't, and she didn't have a very good life. She aged very quickly. They've noticed that with monkeys that they've done too. So some other animals have the ability to actually regenerate and to differentiate cells by dividing and then redifferentiation. So like a starfish, you can actually cut it, and it will grow new limbs and everything. It will actually to be able to regenerate itself on its own. We can't do that. Yeah, iguanas will do that. Geckos. Yeah, yeah, yeah. Like if a predator grabs it, yeah, they eat the tail and they can run away. It's supposed to be pretty gross. Yeah, I had geckos when I first started teaching too. Yeah, and you didn't want to disturb the tails because and they're big too. Yeah, that's where they store a lot of fat reserves in food. Yeah. Jeez. I don't want to see the video. So, so okay. So now cloning. Little mini me photo up there. So what's going on with cloning? So back in 1975, a man named Gurdon came up with the nuclear transfer. This two-step process uses fine needles and a powerful microscope. What he did is he took the nucleus out of a frog egg, um, created an unnucleated oocyte, so that's an egg with no nucleus. He discovered that egg with no nucleus wouldn't divide or differentiate even when fertilized. So, which isn't a surprise. The cell can't do anything without a nucleus, right? So Gurdon's second step surprised many. He moved the nucleus from the frog cell, from the other frog cell, into that egg cell with no nucleus. Okay? These nuclear transfer cells behave a lot like zygotes. They started to divide and divide just as they were supposed to. Until a great ball of cells was produced, just like we would expect. Then this ball differentiated. So actually, it started to reproduce. It's like grow and develop. I just heard it's really messy. Oh, I see. Oh, yeah. And so what happened is these cells differentiated and made tadpoles. All right. And what they were is these tadpoles were genetically identical to the nucleus of the adult frog that they took them from. Okay. It's trial and error. Like this guy was just screwing around. So what they did is they took an egg that was unfertilized and they destroyed its nucleus. They then took skin cells off a frog and isolated the nucleus. Yeah, and they put that nucleus of the frog into that egg with no nucleus, and it started to grow. Yes, these tadpoles will be the exact same as that frog. Yeah. Now, they could actually become a little bit different because differentiation is based on the control of gene expression, and gene expression is kind of controlled by your environment and all these different things. Okay? So, like, your genetic information is the same, but if you lived in a completely different climate and environment, your genes might express a little bit different, and you might look a little bit different. But otherwise, genetically, you'd be the exact same. Kind of cool, eh? I think that's neat. Something to think about. All right, so let's talk about Miss Dolly the sheep. This one was probably the most famous. All right, it was one of the first. So they used cells from an adult sheep's mammary gland from a donor's nucleus. Okay, the cells were grown in a tissue culture. So it was a solution. Yeah. Yeah, they'll put it into a fetal sheep. Yeah. So they took tissue cells from one sheep, Dolly, or Dolly's mom, or donor. They took these mammary gland cells and they grew them a little bit. And then they put that, once they put the cells in the egg, they put that egg in a completely different sheep. So Dolly was the same as this other sheep over here, but she was born from this sheep over there. Yeah. Okay. So Dr. Campbell, what he did is he starved them of vital nutrients, and the cells ceased to grow and divide. These mammary glands, they became dormant. So what they did is they created Dolly through now what's called nuclear transplantation. So using methods like the ones from 20 years ago with Gurdon and the Fogs, technician Bill Ritchie, who worked with Dr. Campbell, took the nucleus from one of the eggs. That's what an oocyte is an egg. That was accumulated from a Scottish blackface female sheep. The Scottish blackface is a breed of sheep common in Scotland and recognized by its black face. That's what they call it. Yeah. So here's the black-faced mom, or the black-faced sheep that delivered Dolly. So what they did is they took Dolly, who was a white-faced sheep, and placed her in a black-faced sheep. Okay. You got your Topanga? And Dolly was born. So the mammary glands were placed into the unnucleated egg. All right. Just hold on. I think this messed. Yeah. So that was Dolly. Now, do I have anything on her here? So yeah, this is Dolly. She was really famous. This is like 97, 98. Did I blow by the ear here? Okay. So yeah, she was born to this black-faced mother here. Genetically had nothing to do with her. Okay. But Dolly did not live a very good life. She aged very quickly. She developed arthritis, and I think she died after about 4 years. So it wasn't a happy time, but it was still. This is probably the first one of the most successful clonings that we had at that time, for sure. Okay. I haven't seen what's been done recently, but I know like monkeys have been done, dogs have been done, most other mammals and stuff have been done. Plants all the time. Plants, that's so it happens quite a bit. Nothing that's been documented. There was an Italian scientist a while back that said he had done it, but then it was found out he was just lying. So. Oh yeah. Now the question is, if you could clone yourself, what would you do with your clone?
I wouldn't clone myself.
No, you wouldn't?